Welcome to the deep dive. Today, our mission is taking a look at something pretty fundamental but complex, the structure of eukaryotic cells. We're diving into Prescott's microbiology to pull out the key ideas for you.
Right. Think of it as your guide to the internal architecture of everything from yeast to algae.
Exactly, because if you've ever felt lost in, all the organelles and what they do, this is hopefully going to clear things up.
And it's crucial because while bacteria often steal the microbiology spotlight, eukaryotes, protists, fungi, they represent this huge leap in structural complexity. Understanding that structure is key to understanding their roles, good and bad.
And we're kicking off with a really striking example of that impact. The blue stain fungus, Grosmania clavigera, it partners with the mountain pine beetle, and together they cause massive devastation in pine forests. You see those red trees in the Rockies? That's often them. Red means dead.
It's an incredible mutualistic relationship. The beetle carries the fungus into the tree. Now, the tree's defense is this toxic resin.
Right, tries to pitch the beetle out.
Exactly. But the fungus, it actually degrades that resin,
Yeah.
uses it as food, so it basically neutralizes the tree's main weapon, protecting the beetle.
Okay, so the fungus runs interference for the beetle, allows it to get established, lay eggs, but then the fungus itself deals the killing blow.
That's right. Once it's inside, the fungus grows these dense masses of filaments.
Mm-hmm.
These aren't producing some chemical toxin. They are physically tough.
So like physically plugging things up?
Precisely. They literally clog the tree's vascular system, its circulation. The tree can't transport water or nutrients. It starves and dehydrates. It's a death caused entirely by a structural blockage.
That's incredible, and it really highlights what makes eukaryotes different, doesn't it? Their potential for a larger size, specialization, and the big one, compartmentation.
Compartmentation. That's really the key concept for this whole discussion. Eukaryotes use internal membranes to create distinct compartments, organelles.
Which means they can do lots of different things at once.
Exactly. They could be synthesizing proteins here, breaking down waste over there, generating energy somewhere else, all simultaneously, without the processes interfering. It's this internal organization that allows for their complexity.
Okay, let's start unpacking those compartments, beginning right at the edge. The plasma membrane. We know bacterial membranes have phospholipids. What's different in eukaryotes? What makes their membranes more robust?
Well, they add some key molecules. Eukaryotic membranes have a lot of sterols, think cholesterol in animal cells, or ergosterol in fungi, and also things called sphingolipids.
And these make the membrane.
They pack together much more tightly than just phospholipids alone. This makes the membrane less fluid, more stable, and helps regulate what gets across. It's crucial for dealing with stresses, like temperature changes.
Interesting.
And it's not just uniform. These lipids aren't evenly distributed. They form specialized little patches, kind of like functional zones on the surface, called lipid rafts.
Lipid raft.
Yeah, these microdomains. They seem to be really important gathering points for proteins involved in things like cell signaling or bringing specific things into the cell through endocytosis.
Okay. What about cell walls? Fungi and algae often have them, but they're not like the peptidoglycan in bacteria, right?
Not at all. Chemically, they're generally simpler, but still very strong. Fungal walls, for example, are typically made of chitin, the same stuff in insect exoskeletons and glucan. Very rigid.
And algae.
Algae often have layers of polysaccharides like cellulose and pectin. Sometimes they even add minerals like silica for extra strength. It gives them very different properties and allows for diverse shapes.
So different structure, different materials, and for getting things in, besides diffusion, they have that special trick. Endocytosis, we'll circle back to that, but let's move deeper inside. Past the membrane, into the cytoplasm. What provides the internal structure? The cytoskeleton.
Right, the cytoskeleton. It's not just static scaffolding, it's a dynamic network. Think of it as the cell's internal framework, its muscles and its highway system, all rolled into one. It's made of three main types of protein filaments.
Okay, what are they?
First, the smallest ones, actin filaments, or microfilaments. They're about 4 to 7 nanometers thick, made of actin protein. These are the dynamic players involved in changing cell shape, movement like amoeboid movement pinching the cell in two during division, and also in endocytosis. They work with a motor protein called myosin. They're like the cell's muscles.
Got it. Muscles. What's next?
Then you have intermediate filaments, or IFs. They're a bit thicker, around 10 nanometers, and they're primarily for strength and structure. They're strong but flexible, kind of like ropes or cables.
So pure structural support.
Pretty much. A key example is the nuclear lamina, a meshwork just inside the nuclear envelope that supports the nucleus's shape. But here's an important point. Fungi and plants generally lack intermediate filaments.
Oh, really? How do they manage?
They tend to rely more on their rigid cell walls and internal pressure for structural support.
Okay. And the third type, the highways.
Those are the microtubules, the largest of the three, about 25 nanometers wide, like hollow tubes. They're built from proteins called alpha and beta tubulin.
And their job?
They have several big roles. They form the spindle apparatus that separates chromosomes during cell division, and crucially, they act as tracks, like railway lines, for intracellular transport. Motor proteins, specifically chytin and dynin, walk along these microtubules, hauling organelles and vesicles around the cell. They also form the core of cilia and flagella, which we'll get to.
That internal transport system sounds vital. So let's follow that path. How does the cell actually make and ship materials? This involves the secretory and endocytic pathways, starting with the ER, the endoplasmic reticulum.
Right. The ER is like the cell's main factory and workshop. It's a network of membranes. You got the rough ER, the RER.
Rough, because it's studded with ribosomes.
Exactly. Those ribosomes synthesize proteins that are destined either to be secreted out of the cell or inserted into membranes. Then there's the smooth ER, the SER. No ribosomes.
And its function?
It's the main site for lipid synthesis, including the lipids needed for making all the cell's membranes. It also plays roles in detoxification in some cells.
Okay, so RER for proteins, SER for lipids. Where do these products go next for sorting and packaging? That's the Golgi apparatus, right?
Correct. The Golgi apparatus, sometimes called the dictyosome in plants and lower eukaryotes, looks like a stack of flattened membrane sacs called cisternae. It has polarity, a receiving side, the cis face, usually near the ER, and a shipping side, the trans face.
Like a cellular post office?
That's a great analogy.
Yeah.
It receives proteins and lipids from the ER, modifies them, maybe adding sugars or other groups, sorts them, and then packages them into vesicles for delivery, either to other organelles or for secretion outside the cell. It's crucial for things like making complex surface structures, like scales on some protists.
With all this production, especially protein synthesis in the RER, there must be errors sometimes. What about quality control? What happens if a protein gets misfolded?
Yeah, the cell has a very sophisticated system for that. Misfolded proteins that come out of the RER get tagged. A small protein called ubiquitin gets attached, usually multiple copies.
Like a label for disposal.
Exactly. This ubiquitin tag is a signal for destruction. The tagged protein is then escorted to a structure called a 26S proteasome.
And the proteasome is?
It's this huge barrel-shaped protein complex. Think of it like a molecular shredder or a wood chipper. It grabs the ubiquitin tagged protein, unfolds it, threads it inside, and uses ATP energy to chop it up into small peptides, which can then be recycled. Very efficient waste disposal.
Incredible. Okay, let's flip back to bringing things in endocytosis. You mentioned it earlier. Can you break down the main ways eukaryotes engulf external material?
Sure. There are a few key mechanisms. One major way is phagocytosis, literally cell eating. This is for engulfing large particles like bacteria or cellular debris. The membrane extends out and surrounds the particle, forming a large vesicle called a phagosome.
Okay, big stuff. What about smaller, more specific things?
Right. For more targeted uptake, you have mechanisms often involving specific coat proteins. Clathrin-dependent endocytosis is a classic one. The protein clathrin forms a cage-like structure around patches of membrane, pulling them inwards to form vesicles containing specific molecules that bound to receptors, like hormones or nutrients like iron.
So very selective.
Yes. And then there's also caveolin-dependent endocytosis. This uses tiny flask-shaped pits in the membrane called caveolae, coated with the protein caveolin. These are involved in transporting small molecules and also seem to play roles in cell signaling.
So phagocytosis for bulk, clathrin and caveolin for more specific uptake. Where do all these incoming vesicles deliver their cargo for breakdown?
They ultimately deliver their contents to the lysosomes. Or, in many microbes and protists, similar structures might be called digestive vacuoles or phagocytic vacuoles.
And lysosomes are like the cell's stomach?
Sort of, yeah. They contain a whole battery of hydrolytic enzymes that break down proteins, lipids, carbohydrates, nucleic acids. And critically, they maintain a very acidic environment inside, around pH 3.5 to 5.0, which is optimal for these enzymes.
How does this stuff get there? Does the phagosome just merge with the lysosome?
Phagosomes typically do fuse directly with lysosomes, but vesicles from clathrin or cavulin pathways usually go through intermediate compartments, first early endosomes and then late endosomes before fusing with the lysosome. It's a more regulated delivery route.
And lysosomes aren't just for external stuff, right? They handle internal recycling, too.
Absolutely. That process is called autophagy, specifically macroautophagy. If an organelle, like an old mitochondrion, is damaged or no longer needed, the cell can enclose it within a double membrane, forming an autophagosome.
Kind of walling it off internally.
Exactly. Then this autophagosome fuses with a lysosome, and the contents are digested and recycled. It's essential for cellular maintenance, especially during stress or starvation.
Okay, fascinating logistics. Let's shift now to the command center and the power plants. First, the nucleus.
So nucleus, yeah. Usually the largest organelle. Its defining feature is the nuclear envelope, which is actually two membranes, an inner and an outer, and the outer membrane is continuous with the endoplasmic reticulum.
And inside holds the DNA. How is it organized? It must be incredibly long.
It is. The DNA isn't just floating around, it's tightly packaged. It's complexed with positively charged proteins called histones. DNA wraps around clusters of these histones.
Forming those beads on a string.
Precisely. Those beads are called nucleosomes. This organization, forming chromatin, allows meters of DNA to be compacted into the tiny nucleus. And getting things in and out like RNA or proteins needed for replication happens through large complex channels called nuclear pore complexes that span both membranes.
And within the nucleus, there's often that dark spot, the nucleolus. What's its specific job?
The nucleolus is specialized. It's the site where ribosomal RNA, or RNA, is synthesized, and where the ribosomal subunits begin to be assembled before being exported to the cytoplasm.
Speaking of ribosomes, you mentioned eukaryotic ones are bigger, 80S versus the 70S in prokaryotes. And their location matters, right? RER versus free.
That's right. Just to recap, ribosomes on the RER make proteins destined for secretion or insertion into membranes. Ribosomes floating free in the cytoplasm make proteins that will function within the cytosol itself, or maybe go to the nucleus or mitochondria.
Okay. Let's talk mitochondria and their relatives. These energy-related organelles, the MROs, they share a common ancestry, right? From endosymbiosis.
Yes, the prevailing theory is that they all evolved from a single endosymbiotic event where an ancient eukaryotic ancestor engulfed an alpha proteobacterium. The classic example, of course, is the aerobic mitochondrion.
The powerhouse of the cell.
Indeed. It's got two membranes. The inner membrane is highly folded into these structures called cristae. This vastly increases the surface area for the electron transport chain and ATP synthesis through oxidative phosphorylation. And inside the central matrix, they still have their own DNA and ribosomes, remnants of their bacterial origin.
But not all eukaryotes live in oxygen-rich environments. What about the anaerobic ones?
They often have modified MROs. A key example is the hydrogenosome, found in some anaerobic protists. These also typically have a double membrane, but they often lack cristae and the electron transport chain.
So how do they make ATP?
They use fermentation instead of respiration. And significantly, one of the byproducts of their metabolism is molecular hydrogen, H2 gas, along with CO2 and acetate. This reflects adaptation to life without oxygen.
Hydrogen gas, wow. Okay, and the other major energy player in many eukaryotes? Chloroplasts.
Right, the sites of photosynthesis in algae and plants. They're also typically bounded by two membranes. Inside, you have another membrane system, the thylakoids, where the light-dependent reactions happen, capturing light energy. These thylakoids are often stacked into structures called grana.
And the carbon fixation, the dark reactions.
That happens in the fluid-filled space surrounding the thylakoids called the stroma. And chloroplasts also have their own evolutionary story involving endosymbiosis, sometimes even multiple rounds leading to the diversity we see.
So much internal complexity. For our final segment, let's look outwards. How do these cells move themselves around using external structures like cilia and flagella?
Cilia and flagella are essentially motility organelles. Cilia are typically short, maybe 520 micrometers long, and numerous, covering the cell surface. Flagella are much longer, 100, 200 micrometers, usually just one or a few per cell, and they move in a whip-like fashion.
Different size, different motion pattern, but structurally similar.
Structurally, they're almost identical internally. The core structure is called the axoneme. It's a cylinder of microtubules surrounded by an extension of the plasma membrane. And the arrangement of microtubules is highly conserved.
The famous 9+2 pattern.
That's the one. 9 pairs, or doublets, of microtubules arranged in a circle around 2 single microtubules in the center. This whole structure is anchored in the cell by a basal body, which actually has a 9+0 pattern.
It sounds incredibly precise. How does it generate movement?
It's powered by ATP hydrolysis. Attached to the microtubule doublets are motor proteins, specifically dynein. These dynein arms reach out and walk along the adjacent doublet.
Causing them to slide.
Exactly. But because the base of the axoneme is anchored by the basal body, the sliding movement is converted into bending. This coordinated bending creates the motion.
And the motion is different for flagella versus cilia?
Yeah. Flagella typically generate planar or helical waves that push or pull the cell through the fluid. Cilia beat with a more complex two-phase stroke, a stiff, propulsive, effective stroke, like an oar, followed by a flexible recovery stroke where the cilium bends to reduce resistance as it resets. Very coordinated.
Amazing. Well, that brings us pretty much to the end of our deep dive into the really remarkable engineering of the eukaryotic cell. We've seen how compartmentation allows for specialization, how the cytoskeleton provides internal structure and transport, the vital logistics of the ER, Golgi, and endocytosis.
And the fascinating evolutionary story embedded in the energy organelles like mitochondria and their relatives. It's a huge jump in complexity from prokaryotes.
It really is.
But, even with all this structural difference, it's worth remembering the deep molecular unity underneath it all. The fundamental genetic code, the core processes of DNA replication, transcription, translation, many basic metabolic pathways. They're remarkably similar across bacteria, archaea, and eukaryotes.
That's a great point. Structural emergence built on a shared molecular foundation. So, thinking about that evolution, here's a final thought for you, the listener, to consider. We talked about mitochondria originating from endosymbiosis and anaerobic relatives like hydrogenosomes. How might studying the full diversity of these mitochondrial-related organelles, the MROs, give us clues about the actual environmental conditions, the selective pressures that were shaping early life back when oxygen was rare on Earth?
That's a fascinating question. Using modern cell structure to probe ancient environments.
Exactly. Something to think about. Thank you for joining us for this deep dive.
Yeah, thanks for listening.
We'll see you next time.